All right, folks, let's do this last video in this series of 5 videos about the cell. And in this one, we're going to be looking at intracellular structures that are not part of that endomembranous system. So what does that mean? That means these structures are inside the cell, but they do not have that phospholipid bilayer, so they cannot be fusing with any of those other structures. And the first one we're going to talk about is the mighty mitochondria, the site of ATP production. That's its job. It's converting a chemical energy into stored energy. So here's what I want you to do, because I know you know it. I want you to go back and think about cellular respiration, the 4 steps, or like the 3 main processes plus the linking um, equation. I want you to think where in the cell those things occurred, where they happened, and before you move on to the next slide, just review them really quickly in your head. When you look at this drawing of the mitochondria on the left, at first you might think, well, Dr. Powell, it looks like it's got 2 layers. Like it might have a phospholipid bilayer. Yeah, it has 2 layers, but it's not your phospholipid bilayer that's going to fuse with anything else. In fact, what's really cool is the mitochondria actually used to be a prokaryote of its own. So yeah, it's got the right kind of thing, but it is independent. In fact, it makes mitochondrial DNA, does its own thing. Okay. And when we look at that inner membrane, it has those infoldings known as the cristae. That's where those proteins were for that electron transport chain. And then it's got the lake in the middle known as the mitochondrial matrix. That's where the linking reaction as well as the Krebs cycle was going to occur. And then if you look at this picture, what type of microscope would this be? And specifically, what type of electron microscope? That's right. You've got it. So this oval thing would be the mitochondria. And you can see all the lovely little cristae going across. Now look next to it. Do you see these little things with all the little polka dots on the outside of it? Each one of those little tiny polka dots is a ribosome. And so and the ribosomes are studying the outer membrane of this structure. So what structure do you think is that's studied, that's all rough with those little ribosomes? Isn't that so cool? Yes, that's rough endoplasmic reticulum. Okay, so we're done with mitochondria. Moving on, let's talk about cytoskeleton. Cyto meaning cell. So this is microscopic skeleton within the cell. It has nothing to do with bones. It's a network of protein fibers. Generally, we can classify it 3 different types: intermediate fibers, microfilaments, and microtubules. Mostly in this class, we're going to be talking a lot about microtubules today. Here it's showing like differences between the 3 main types. I'll be showing you specifically about different types of cytoskeleton when we talk about it. Different proteins are involved. You may know about keratin already. Yeah, that's the same keratin that's in your hair and that's in your nails. That's an example of a protein fiber in cytoskeleton. Other examples would be actin. Actin also works with myosin, which gives you muscle contraction. So that gives us functions of cytoskeleton, which are quite a few. So yes, cell movement as in muscle contraction, giving shape to a cell. If you look at this picture in the bottom right, do you see those red fibers on the outside giving that shape to that cell, being kind of square-ish? If you go back and look at those pictures from the Henrietta Lack slide, you can see a lot of the cytoskeleton in those pictures. We also have green ones here and yellow ones in the other cells. They can form, especially the intermediate filaments, basically can form like roadways so that organelles have to travel. You know how like when you're going down like a boulevard and it's divided and you have to go all the way down here and then you turn to come back. Yep. They make organelles do the same thing. They're also going to be important in cell division. I'll show you that shortly. And they can anchor structures, including anchoring the cells so cells don't pull apart from each other or apart from other tissues. So this is the part of the organelle that has cytoskeleton that is involved with cellular division, and specifically centrioles. And what centrioles are, this organized microtubules. If you look at them, we have like these triplets. We have 9 sets of triplets that form like this little ring, and we've got 2 sets of rings that are perpendicular to each other. And they form this little structure. These 2 that are perpendicular to each other with a little bit of attached matrix form this thing called a centrosome. And what happens is this is involved when the cell is dividing. So when we are talking about mitosis and meiosis, this is going to be involved. And so we have the little centrioles at each end, each pole of the cell, and then from them, they are going to send microtubules shooting out from them that are going to be attaching to the chromosomes, which are lined up in the middle of the cell. And then what's going to happen is, and that is called a spindle. And then what's going to happen is those spindles are going to be pulling the chromosomes back towards like the north and the south pole of the cell, or the east and west pole, whatever you want to call them. They pull them to opposite poles of the cell. And so that's going to be important in cellular division. Another structure that involves cytoskeleton is something called cilia. And a plural, cilia is the plural form, and the singular form is cilium. So here is one cilium, and as you can see, it's got a whole bunch of microtubules, and they're arranged differently than they were in the previous slide. And what cilia do is they create external currents. So pretend you're a cell and stick your arms up in the air and move them forward, and then whoosh, back, and then move them forward gently and back and forth. And if you were in the classroom, I'd have you moving them back and forth. And what happened is you would start feeling an air current, because that's what cilia do. They create these external currents. That's their function. Create these currents so as they can move substances outside the cell. So if it's air, it's an air current they're making. Lots of places in your body we can have mucus, in which case they are going to create movement of that mucus along. So for instance, at night when you were sleeping, I don't know if you realize this, but your nose and your airways, those cells are coated with this mucousy substance, and all night long, your cilia are there creating these currents. Here's these lovely little cilia. Do you see how many thousands you have on each cell? Aren't they really kind of cute? And they're creating these cilia, and they're moving whatever inhaled pollutants, particulate ash, if you're by a fireplace, whatever it is, they are bringing those up all night long. Now, they're also in other places in your body, such as ladies in your fallopian tube, and their job is to help bring that oocyte, that thing that you call your egg, the oocyte, so it can meet Mr. Spermy and make a baby. Okay. But here's the thing, ladies and gentlemen. If you smoke, you destroy your cilia. How do we know this? What's the first thing a smoker does every morning? Right? They have a smoker's cough. They are hacking up everything out of their lungs. Non-smokers don't have that morning smoker's hack. Why? Because their cilia were working all night long, like a little elevator. In fact, it's called the mucociliary elevator, to bring all that stuff up out of your respiratory tract. And that smoking not only destroys the cilia in your respiratory tract, it destroys the cilia in your reproductive system, which is why it's harder to get pregnant, ladies. And that is not only if you're the smokers, also if you're having sex with somebody who is a smoker, and that also includes oral sex. And I'll just leave it at that and just move on. To let you know, here's another what my children used to call useless piece of trivia, which they would always throw at me when I would ask them, so how'd you do on that test today? They would throw out a useless piece of trivia. In other words, let's change the subject. Did you know that some unicellular organisms have thousands of cilia? They have so many that they can create currents like in the fluid that they live in that can actually move the organisms. But remember, cilia are not capable of moving human cells. They only create external currents to move extracellular substances in humans. There's only one human cell that can move, and that is the sperm cell, and it can move only after it's outside because it's a lazy cell as long as it's inside the male, and because it doesn't do work while it's still inside. But once it's outside the male, then it uses energy to move its flagellum, and the flagellum can move because it also has cytoskeleton. It does not move back and forth like your cilia do. Instead, it has a sidewinder kind of movement. So a whip-like movement like a sidewinder snake. Now there's one last structure I want to talk about that does not have any cytoskeleton in it, but it also is a modification of the surface in that instead of having a flat surface, they are microscopic cellular extensions like the finger. So instead of the cell being flat and square like that, you would have finger-like extensions. Now these finger-like extensions are called microvilla. Think microvilla, and that's because they are short like this instead of long like your cilia. A cilium is 20 times longer than a microvillus, so only 5% the length of the cilium. So they can only be found on a surface of a cell that is not attached to any other cell or attached to any other substance. So only on that free surface. And here's why we want to have microvilli. Say, for instance, this is your digestive tract and you've eaten a bunch of protein and you want some amino acids. And you're breaking them down, and here go the amino acids. Would you rather have this, let me turn this amount of surface area, or would you rather have this amount of surface area? Do you see how the microvilli give that much more surface area? So we're going to find microvilli when we want to be doing absorption and or secretion. Sometimes the same cell will be absorbing and secreting stuff. Sometimes cells only absorb, sometimes cells only secrete. But the point of a microvillus is to increase the surface area for absorption and or secretion. Okay, so here are two photomicrographs. This one is transmission electron micrograph. You got some beautiful mitochondria over there, and here are the microvilli. Why do you think you have a bunch of mitochondria? Because for absorption, for phagocytosis or pinocytosis, that's going to require energy. Do you see how we build on everything? We are building on earlier today, and earlier today is building on 2 days ago, which is building on last week. And in comparison to that transmission electron micrograph, here is the scanning electron micrograph, so you can actually see the surface with a whole bunch of microvilli there. And then this is a picture showing a surface but with both microvilli and cilia. And inside the cilia, you can see that there is cytoskeleton inside it, but inside the microvilli, you can see the absence of cytoskeleton. So multiple differences between the two. All right. So these are not organelles, but I just want you to realize that outside the cell, in other words, extracellularly, there's more than just water. It's what we call the extracellular matrix. So yes, it's a watery based environment, but there's a mixture of proteins, different types of proteins. There's going to be fibers. There's going to be polysaccharides. You remember what a saccharide was? Sugars. There's going to be a plethora of things that the cell secretes and gets rid of. But they're not necessarily waste products because some of these things will help anchor the cell in the tissue it's located, and other ones are super important for cellular signaling. In other words, for cells to signal to other cells what it is they're supposed to be doing. Cells are sitting around waiting for messages to tell them what to do. And cells are held together by intercellular attachments. So we have that prefix intra, I-N-T-R-A, meaning inside, and extra, meaning outside. This is inter, I-N-T-E-R. Inter means between. So intercellular means it's between cells. So yeah, technically it's extracellular, but it's more specific. It means it's right between cells, okay? Because extracellular could mean it's a mile away, okay? So like if you're in a parking lot, that inter-automobile space in a parking lot is like that foot between, okay? It's not, we're not talking half a mile between cars. And intercellular attachments are real important in our skin. All of our skin cells are attached to each other so that when you rub on them, they're not falling apart. Most of our muscle cells are attached to each other by these. Some of our neurons are attached to each other. We'll talk about these later on when they are important. So thank you so much for all your hard work. I ask you to please stay focused. Make sure you're taking notes. Make sure you're doing some of those techniques. Make sure anytime I ask you to do a little activity, you please do it. Take advantage of all the little extra credits because all the little points add up. And remember, if you're getting points for studying, it's a double whammy. You're getting points for studying, and that's also going to help you when you take your quizzes and your tests. And then I have one last slide for you, and that is, do you know these? Can you name these 11 things now? So before you sit and do it right now, I gave you this picture. Go back, study all this stuff from these 5 slides, then go back and can you name these 11 things? And then can you name their functions as I describe them to you? And if so, then you're ready to move on to take a quiz on this material and you're ready for your test. Once again, thank you so much for all your hard work, and I will see you again soon.